Asexual reproduction in organisms. Now, when it comes to reproduction, it can either be sexual or asexual. So, sexual reproduction involves two individuals, a male and a female. So, you're going to have the male and female gametes fusing to form a zygote. Asexual reproduction, on the other hand, requires only one individual. So, this organism gives rise to a new one. Now, of course, the new organism is going to be identical to the parent. Now, I'm going to discuss three main types of asexual reproduction: binary fission, sporulation, and last one, budding. Let's start with the binary fission. Now, this is a type of reproduction that is seen in bacteria and also in some members of kingdom Protoctista, such as the amoeba and the paramecium. So, binary fission is the process whereby a unicellular organism divides to form two daughter cells. These daughter cells are going to be identical to each other and to the parent. Why? Because they are going to have the same genetic makeup. So, binary fission involves the following steps. In step 1, you're going to have the nucleus dividing into two. So, once this happens, the cell membrane then starts to constrict. Now, it continues to constrict until it cuts the parent cell into two parts. These are then going to form the daughter cells. Now, one thing about binary fission is that it's very, very fast. Let me give an example. If conditions are favorable, when you have necessary nutrients and oxygen and moisture and optimum temperature, what will happen is that you're going to have bacteria dividing after every 20 minutes. So after every 20 minutes, you're getting two new bacterial cells. If this rate is maintained for 12 hours, a single bacterial cell can give rise to over 68 billion cells. It boggles the mind, right? Now, the good news is that these numbers are rarely attained because it reaches a point whereby you're going to have depletion of nutrients. The nutrients that are present are not enough. You're going to have accumulation of waste products which are toxic. So this slows down the rate of cell division. But my point stands, binary fission is very, very fast. Now, moving on to our next type of reproduction, sporulation. Now, let's first define what a spore is. So, a spore is a microscopic reproductive unit. This simply means that, number one, it's microscopic. You cannot see it with your eyes, your naked eyes. And number two is that because it's a reproductive unit, if it manages to land on an area with all the necessary resources, the nutrients and moisture and so on, it can then germinate to form a whole new organism. So, sporulation is a type of reproduction that is seen in fungi and even some species of plants. Now, for this explanation, I'm going to use the example of the rhizopus. So, the rhizopus is a saprophytic fungi. What does this mean? It's saprophytic. It feeds on dead, decaying organic matter. Now, a very good example, which I'm sure almost all of us have witnessed, is the black surface structure that forms on decomposing bread and fruits. That is rhizopus. So it's feeding on dead organic matter, which, for example, in this case is the bread. In the rhizopus, you're going to have the spores developing through mitosis in order to form a structure called sporangium. Now, within the sporangia is where you're going to have the spores. So the spores develop, and when mature, the walls of the sporangia burst open, releasing the spores. So if it lands in an area with all the necessary conditions, it can germinate to form a new rhizopus. Our last one, budding. Now, budding is a form of asexual reproduction that is seen in yeast cells. Remember, yeast are organisms that are members of kingdom fungi. So what happens is that a new organism is produced as a bud of the parent, as an outgrowth. And it's going to be identical to the parent. Why? Because it has the same genes as the parent. Now, before I go into the actual budding process, I will just like us to make a note of the yeast cell. So the yeast cell is surrounded by a cell wall that is made up of chitin. This is a complex carbohydrate. It's actually a polysaccharide. We also have the cell membrane and different cell organelles such as the nucleus, the vacuole, and of course, food granules. Now, when conditions are favorable, when you have enough nutrients, you have moisture, you have optimum temperatures and such, yeast cell undergoes reproduction through budding. So the first step is a small part of the cell wall softens. It becomes soft to allow for the formation of a bud. So the bud then bulges out. This is then followed by the division of the nucleus. So the nucleus divides into two to form two nuclei. Now, the reason why we need to have two nuclei is because at the end of it, you're going to need one nucleus for the parent cell and one for the bud. So as this proceeds on, what happens is that the bud then grows in size and forms new cell organelles that would sustain its survival. Now, once the bud has become self-supporting, now it's now independent, it can take care of its own body processes, it starts to separate from the parent cell. So at this point, you end up having a new yeast cell. Now, I want to say this, the budding process is extremely fast. So essentially what happens is that you're going to have the first bud still forming when a new one is being created. So this ends up creating this chain of cells that will look like this. And that, my dear students, brings us to the end of this lesson.